This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 297, recorded on October 5th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone.
Got any hurricanes down there, Michael?
No, it is actually spectacular weather here in Charleston. As they say, it's Chamber of Commerce weather. It is absolutely gorgeous. There's not an empty table in any of the restaurants. They are
nice.
Packed.
Very nice here in New York today. It is like, right now it's 25 C.
Wow.
And it's sunny. It's just, I was up at Columbia this morning, and it's just gorgeous. Also joining us from St. Louis, Missouri, Petra, Levin.
Hello, it's great to be here. I think we have the weather that is coming your all's way. It's rainy, and it's supposed to drop about 30 degrees. So it was 66 this morning, and
wow.
Tomorrow it's supposed to be in the low 50s.
Ooh.
So.
Thank you for sending that to us.
Well, anytime. Just weather moves west to east. That's just how it goes.
Today, of course, we have a snippet and a paper. You know, on TWiM, we stopped calling them snippets because they're both equally
They're
long.
both papers.
I gave you a shout out on the last TWiM, Michael. We said something about copper. I don't know if you heard it.
I didn't. I'm a bit behind. I had my cousins visiting from Chicago this weekend, so normally I catch up on Sundays listening to TWiM.
What was it about copper now? I don't even remember. I forget the paper.
Now I'm curious. I'll have to go and listen.
Anyway, today our snippet is from Petra. Tell us what you have, Petra.
So today I have on a paper on lipoproteins in Bacillus subtilis spores. So Bacillus subtilis is my first love as a bacterium. My lab still has one project on it, but I don't work on sporulation. But this paper is entitled, "A Family of Spore Lipoproteins Stabilizes the Germination Apparatus by Altering Inner Spore Membrane Fluidity in Bacillus subtilis Spores." It's by Matthew Flores, Kate DeRissi, Shreya Chowdhury, Michael Lau, and David Popham at Virginia Tech and at the Advanced Light and Electron Microscopy Center at the Robert Koch Institute in Berlin, Germany. I like this paper for many reasons. First of all, I like the people who wrote it. But I also like this paper because it talks about bacillus spores, which are actually very similar to spores from... So Bacillus subtilis is a gram-positive, a monoderm. It is a model for studying gram-positive bacteria. It's also a model for understanding development in bacteria, and I'll get to that in a second, but it's related also to several pathogens, including Clostridia difficile, which I think our audience might be familiar with, that causes major problems in the gut, especially after taking antibiotics that wipe out all the bacteria that compete with it. It's also a relative of anthraces, which, we rarely see. I think it's more of a problem people in agriculture, but it's been, bounced around as a bioweapon. But all of these bacteria have in common the fact that they, under specific conditions, and these conditions can vary, form resistant endospores. So Bacillus subtilis is kind of the classic example of this, and sporulation in Bacillus subtilis happens when the cells are starved for nutrients, and they're also crowded because the cells send signals to each other. I'm starving. Are you starving? Basically talking to each other. And when the conditions are appropriate for sporulation, it initiates a developmental pathway that the most obvious morphological hallmark of sporulation is, instead of dividing in the middle to produce two rod-shaped cells of equal size, it puts the septum near one end. So it forms an asymmetrically positioned septum that divides the cell into a small compartment and a big compartment. And the big compartment or big cell is called the mother cell. And the small compartment is called the pre-spore or forespore. In the United States, we say forespore mostly. But what's really amazing is, so it divides and it looks like a normal division, but the peptidoglycan, it's thinned between the two cells, and the mother cell, the big cell, actually engulfs, almost like phagocytosis. It basically wraps its membrane around the forespore until that small cell is essentially floating inside that mother cell. And that process generates two membranes, the outer membrane, which is derived from the mother cell membrane, and the inner membrane, which is derived from the forespor's membrane. I think if you draw this out, it's pretty clear. But you have two membranes in that spore, that pre-spore or forespore that's floating inside the mother cell. And the mother cell, the membrane, the outer membrane of the spore, actually makes peptidoglycan that we call cortex. And the mother cell also makes proteins that go on the outside that are called the coat. And the spore makes special proteins that are stuck in the inner membrane, including proteins that recognize germinants, which I'll get to. And also the spore makes molecules that protect its proteins that are left there, and it dehydrates. So one of the things it makes is called dipicolinic acid, which is chlorinated with calcium, and that actually protects the proteins in the cell. The spore is dehydrated. The DNA is actually around these small acid-soluble proteins called SASPs, and you don't need to remember those. But basically, it becomes a dehydrated cell, and it has this very elaborate cortex and then coat around it. And then the mother cell dies, it releases the spore, sort of altruistic. And that spore, once it's released, is really resistant. You can heat it to 80 degrees, probably boil it briefly. You can treat it with chloroform. Basically, nothing gets in. It's dry, so that also helps it be heat resistant. And it's very stable. People think maybe even millions of years. There's some debate in the field if this is true or not. There's a bacteria called Bacillus thuringiensis, which infects insects. And people have found insects in amber and isolated Bacillus thuringiensis that in theory is hundreds of thousands, millions of years old, and gotten it to germinate. Unclear contaminant or not. But the idea is they're certainly resistant. They certainly last years, decades even. Anyway, so the spore, though, once it sees a whiff of a germinant, which could be just an amino acid or something, and it's in a wet condition, it instantly germinates. And germination involves degradation of that cortex, that peptidoglycan between the outer and inner membrane. And it also involves release of the dipicolinic acid that was kind of protecting the proteins, and hydration of the spore. So just so we understand, right, so we've got two membranes, it's cortex in between, the inner membrane, the outer membrane. We've got a spore with stuff in it. We have proteins on that inner membrane that are basically receptors. So they sense the germinants. And the question that this paper is asking is really looking at how these proteins, again, so these proteins that allow for germination, they have to be active and protected on that inner membrane so that when there is a germinant, the cell can essentially reconstitute itself. It can come out, germinate from that spore and become a living cell again. And this paper actually, and I think very smartly, looks at some proteins that are lipoproteins, so protein lipid kind of hybrid molecules that are well conserved throughout the spore forming group of bacteria, the Clostridia and the Bacilli. And they had previously been identified and found to be important for efficient germination, but not for morphology. So other labs had knocked them out and seen like a decrease in the how well bacteria germinate. So you had germinant and then you plate and you look for colonies. And if you have the same number of spores, you should get a certain number of cells recovering. Not everybody recovers, but it's pretty high. I think it's like 85%. In this case, they saw a somewhat of a reduction, and they were looking at these lipoproteins. And what was really kind of cool and I think intrigued them is not only were these lipoproteins previously identified as being important for germination and also conserved, one of them was actually encoded in the same operon as an enzyme or a gene called SLB that encodes a protein that helps degrade the cortex. So again, there's this peptidoglycan that's pretty thick and around the spore, and it has to be degraded when cells germinate. And so the spore comes with things to degrade it. It can't get it from anywhere else. And so one of those proteins on the inner membrane is called SLB, and there's also CWLJ on the outer membrane that also does it. So it degrades it from inside and outside. But one of these, the SLB enzyme for degrading peptidoglycan, was in the same operon as one of these lipoproteins. So it means that two proteins that are probably important for germination, we know that they're both important for germination, are encoded together. And that kind of signals that they're probably involved or related in some way. So I think the fact that they were there, we also know that lipoproteins, you can think about cholesterol or something like that, these alter membrane fluidity. And membrane fluidity, right, if you heat up lipids, right, you take butter and you heat it, it melts. Same with membranes. If you heat it up, the membranes become more fluid. Membrane fluidity is actually not great for something that's trying to be stable. So the idea, I think this idea that lipoproteins probably keep membranes stable across heat, that this is encoded with something we know is important for germination, and that defects in this were shown to be involved in germination kind of signaled to the Popham lab like, hey, maybe these guys are important for protecting that inner membrane, which is arguably the hardest thing to protect because you can dry things out in the middle of the cell. But again, you have to have these proteins on the outside that are really robust and able, and when they sense the right condition, they have to go. They can't wait. They have to just germinate. So they basically, with this in mind, and this actually, I think, excellent hypothesis as it turns out, they start by doing some genetics. So Bacillus encodes 4 of these lipoproteins, and it encodes 2 proteins, let's check if their names, for germination, GER-A, and then 2 that sort of work together, GER-BK, and they respond to different signals. So we have GER-A, GER-BK, which are the proteins, the receptors for the germinants, if you want to think of them that way. And we have 4 lipoproteins. And they started to do some genetics. The first thing that they did was to knock out all in singly and in combination these lipoproteins. So they make different variations on the theme where they knock out 1, 2, 3, or 4 of these lipoproteins. And then they see if germination is affected. And they do this really carefully. They either add germinants they know activate GER-A, or they look at ones that activate GER-BK. And what they found is that if you have mutations in these lipoproteins and you try to germinate by activating GER-A, you actually are about 2-fold reduced in germination efficiency. And they're not looking at germination efficiency by outgrowth. They're actually looking at a drop in optical density, because you can use optical density in a spectrophotometer, and you can just look at cells. And when the spores germinate and they fill with water, they reflect light differently. And so you can see a drop in optical density. And the drop in optical density in wild type cells is about 70% when you add a germinant for GER-A. But these lipoprotein deficient spores, whether it was one or all of them, were about 2-fold reduced in their loss of optical density, which says they're just 2-fold down for germination efficiency. They're still germinating, but not as many of them, basically.
And the experiment Petra's just describing is beautifully illustrated in their first figure,
yes.
Panel A. I mean, it's night and day, folks.
Yes, it is actually a really beautiful figure, and you can just look at optical density, and it's really clear that even it doesn't matter if they're alone or in combination. If you knock out GER-A, you basically don't germinate at all, which is also really nice to see. You see really no change in optical density. So it's a really great assay. It's a really simple assay. So that is actually excellent. They also look at release of this dipicolinic acid because, again, it's inside the spore, and when the cells germinate, they release it. And they see, in the same figure, they see a difference in release of the dipicolinic acid. That's figure 1C. And my favorite, because it uses a microscope. So the spores, because they're dehydrated, they are phase bright in a phase contrast microscope. So you can literally watch germination because they go from phase bright, they look white in the phase contrast microscope, to dark. And you can see that about half the number of cells in the mutants are germinating, and this is figure 1D. So again, this is a visual. I love a visual. And it's pretty obvious that they're not off. It's not, a lot of times we expect mutants to be on or off. And here it's just not as efficient. Which again, if you think about the hypothesis that these lipoproteins are protecting JUR-A, maybe, the fact that they're gone doesn't completely destroy it. It's not active in a subset of cells. Interestingly, when they look at JUR-BK activation, they don't see anything. So it's only germination through JUR-A. So not JUR-BK, which says that these proteins are really important for JUR-A, but not for the other germination pathway. The next experiment they do is look at heat resistance. So heat resistance is a classic way for selecting for spores. If you wanted to identify spore formers from the environment, you could scoop up cells from the soil, and then you could heat them to 85 degrees C for like 20 minutes, and then you would kill everything in there that isn't forming spores, mostly.
Like you said, soil. Perfect.
As opposed to dirt.
Yeah.
I never say dirt. I just realized that. Okay, yeah, soil.
Soil, yeah,
not dirt.
soil.
I don't know what would
dirt.
happen with dirt. I feel like they're this. Anyways.
They are the same. It's just that the agronomists always make you talk about soil.
Oh, okay. I think I was raised.
You get
with
into
a really
trouble
big
when
garden,
you call it dirt.
So maybe that's it. But anyways, if you take the soil, which just sounds so much richer too, right? If you want to isolate aerobic spore formers, take the soil, heat it to 80 degrees for 20 minutes, and then plate it. And pretty much that's all you're going to get out. In any case, the ability to withstand 85 degrees over time, it was reduced in strains. Not if you're only lacking the lipoproteins, actually it didn't make much difference. But if you're lacking the lipoproteins and CWLJ, so one of these enzymes for degrading the cortex, but the one that's on the outer membrane, then they saw a reduction in viability at high temp, as if somehow without that enzyme and without the lipoproteins, now JUR-A might be more sensitive. And they have this picture. It's actually a beautiful set of genetics, is figure 2. They were curious also whether the lipoproteins could complement one another. Because again, the lipoprotein in a way is a lipoprotein. And they find actually that they can complement one another, but you have to have at least 2 different or 3 different kinds. You can't just overexpress one and have it do the job of all 4. And that is figure 3. And that's actually interesting because it says that there's some kind of interactions going on that might be beyond just membrane fluidity. They originally speculated that membrane fluidity was actually what's happening here. Again, if you have a higher melting temperature, you would be more protected over time, especially at high temp. And so they actually looked directly at this. And I actually was not familiar with this assay, but it's really quite nice. So they use a fluorescent dye called Laurdan, Laurdan. And that dye is lipophilic, so it goes to the outer membrane and the inner membrane. But it turns out if you take off the spore's coat, so outside the outer membrane is this specialized coat, and you can decoat the spores. People know from other things that outer membrane goes away. So now when they stain with the Laurdan, they know they're only looking at the inner membrane. And they see a shift in the emission spectra of Laurdan when the fluidity changes. And when they do that, and this is table 1, they see that in mutants lacking the lipoproteins, especially lacking 2 or 4 of them, they see a change at 25, but they see a sort of more, they see less of a change at 42 degrees. But the point of that is that altogether, this test indicates that these lipoproteins are doing what lipoproteins do. They're stabilizing the membrane. And so altogether, this model suggests that these lipoproteins are there, that they're reducing membrane fluidity and protecting the spores from heat. What's a little confusing, and especially since they can't just overexpress one to get it to do the job of all four, is that they only see protection for JUR-A, not JUR-BK. And, we can speculate about why. It could be that some of these are actually interacting specifically with JUR-A, and that change in membrane fluidity is not as generic as it appears. It could be that really it's maybe also serving some kind of chaperone protective function directly. What's also interesting is even though the membrane fluidity definitely changes, they don't see much of a change or any detectable change in the architecture of the spore. And if you look in if you want to know what a spore looks like, there's some really pretty electron micrographs in figure 5 where you can see the spore and you can see the coat and then the cortex, and you can clearly see the inner membrane. The outer membrane is basically not visible ever. The coat is 2 layers, and then the cortex is so the coat is like dark layer and then a lighter layer, and then the cortex is gray. You see the inner membrane, and then you can see inside the cell. But there's really no difference. Like I can't see any difference between those two either. So I think this is really interesting, and it's another. The spore has these different layers, and each layer is important for it to be resistant. And so we've just uncovered another sort of addition, many different things that kind of help protect it. And I guess the way I kind of was thinking about this when I was going through these yesterday was like, it's like layering. It's like the original layered outfit is actually the spore, right? You've got the coat that adds one kind of protection. You can almost think of that like the shell on your jacket. And then maybe you have a fleece, right? And that would be another layer. And then maybe you have a nice wool undershirt that might be this inner membrane in some way to kind of keep you protected from the temperature. So I think that I like that kind of way of thinking about the spore. And I really like this because they had a hypothesis and they tested it and they did a really rigorous job. But it also gives you like, what are these lipoproteins doing? People knew they were there. But it's like, oh, that makes so much sense what they're doing. But it also leaves you with a mystery, like what the heck is going on with JUR-BK? So I'm really curious about that.
You wonder if their next experiment is going to be using some of this AI software to ask how these proteins interact in the membrane itself to see if the AI could figure out who's talking with whom.
I've never asked AI that, but that's a question I have for many proteins. So it would be good to know. I think my next experiment would be to give a student JUR-BK and be like, okay, so what's protecting JUR-BK? Yeah.
So, Petra, are spores living?
Oh, you had to ask that.
Yeah, spores are living. They're just dormant. It's like our seeds living. Seeds are living. Viruses are hard, though, because, again, they can't live unless they're unless they become parasitical, right? Yeah, whereas these guys, all they need is a little germinant, and they're back to normal.
So what's the minimum needed for germination of a spore?
Water? No, they need water and an amino acid. Water alone won't do it. They need L-valine, I think, in this case, or
yes.
Oh,
yeah, no. So you can put them in water and nothing happens. But if you give them the right amino acid.
A seed, too, you add water. I think you need soil for a seed, right?
No, they'll do it in, I think, water and trace because seeds carry all their nutrients right with them. You've seen people do that. Kids always do this where you take a paper towel and a seed.
We've all had bean sprouts.
Bean sprouts, right, exactly.
Right.
A spore won't do that. That's how you can do that heat kill assay because that's literally done by doing serial dilution into test tubes in water, and then you heat it up on a heat block, and then you plate.
So, a virus, if you added water, it wouldn't do anything.
No, exactly. It
has to get into cells.
If you can add water, it's like these are like those monkeys, those sea monkeys you can buy in the store, right? Yeah, exactly.
In fact, we were talking about this the other day, sea monkeys. Yeah.
Sea monkeys? You must have had Dixon in the house.
Basically, they're are they brine shrimp?
Yeah, they're brine shrimp. They're very
add water.
disappointing, but if you think they're going to look like the picture on the box, not at all.
Yeah. No, but that's why I say a virus particle is not alive. But if it gets into a cell, then the infected cell is alive. So that's a two-phase organism where you have. But spores, and I use spores or seeds as a counterpoint. I think
that's a good counterpoint. Yeah, a seed is even better though in a way. Well, I think a spore is great, but the seed's carrying its like nutrition with it.
So a spore is sitting in soil, Petra. And if it encounters some water and an amino acid, it will begin to germinate. That's it.
Yeah. So in any population of Bacillus, my understanding is there's always some low level of sporulation. So Bacillus, for example, subtilis, one of the first things that we tell students who work with it is don't put it in the refrigerator, especially if you come from E. coli, you just shove your plates in there, which is not necessarily great, but Bacillus we leave on the bench where it becomes extremely stinky. But the reason for that is if you put Bacillus in the cold at 4 degrees, so 40 degrees Fahrenheit, basically, it lyses and it's disgusting. But which always begs the question, like, how the heck did this thing survive out in the wild? And it's probably that there's some low level of sporulation all the time.
I see.
I mean, sporulation is absolutely fascinating. I often tell the story of I was assigned to grow up a bunch of Bacillus megaterium because it makes such large spores for an undergraduate laboratory where they had to perform a spore stain. And so I growing it up, and it was in the warm room on a shaker, and it was vigorously being aerated. Megaterium is one of these obligate aerobes. And as I took the flask off of the shaker in the warm room and walked back to the lab to dispense it into tubes, because it's so sensitive to the oxygen concentration, as the oxygen tension dropped in that maximum growing flask, the entire flask cleared. They all just lysed synchronously. And I had an entire, 3-liter flask full of spores rather than megaterium.
That's amazing.
And it was wild.
Because subtilis likes oxygen, but I've never seen it do that.
Megaterium does it. It was wild. I still remember that day, pulling this flask, and I was supposed to dispense it in tubes. I go to my mentor and I say, "What happened?" He says, "You weren't shaking it as you were walking." And I said, "What?"
It's funny. Thank you, Petra.
Yeah.
So, Michael, it was a story that we did on TWiM. Cats have a retroviral envelope that mediates copper homeostasis in them.
Okay.
It's so copper is pumped into cat cells by a transporter that is the receptor for the retroviral envelope, and the envelope plugs it and mediates copper transport. Isn't that amazing?
That's amazing.
So copper is such an important trace element that I think Mother Nature, because it's involved in redox enzymes, and redox is so important to the cells, controlling everything. I think Mother Nature has figured out these precious trace elements, and there's not that much copper, just floating about in the environment. It's not like iron.
So whenever copper comes up, we mention you, Michael. You're the man.
Well,
of copper.
we're gonna have. I'm gonna suggest a paper on antimicrobial copper screens. They have developed transparent copper screens in which they demonstrated the antimicrobial activity. So from all of us who use touchscreen devices, and it's in Nature Communications, and I just got a note from the editor that it was accepted, and I was one of the reviewers. I'm sort of outing myself, but it was a fascinating paper.
Okay. So, Michael, what do you have for us?
I have something completely different, and the title of the paper is Sub 1.4 centimeters cubed capsule for detecting labile inflammatory biomarkers in situ. That's the operative word, in situ. It's authored by 23 individuals from the synthetic biology group at MIT. And the first author is Inda Webb, Jimenez, Lou, and it goes on for the 23 authors. The first thing you may wish to do before you delve into today's paper is, as a prelude, you may want to review the movies from 1987 and 1966, respectively, titled Inner Space, that starred Dennis Quaid and Martin Short, and then Fantastic Voyage, starring Raquel Welch and Stephen Boyd. Unfortunately, you're going to have to rent them, as none of the streaming services in the U.S. seem to be offering you an opportunity to view them without specifically renting them. From that, I hope you get the idea that today we're going to find ourselves on our own fantastic voyage, a trip where we're all going to need to recall the disciplines of microbial physiology, molecular genetics, the wonders that our reporter technology can offer in deciphering complex problems, especially as the reporters can now emit light. In the old days, they just turned blue, but now they can emit different colored lights. Some very sophisticated bioengineering with a smattering of biochemistry, reactive immunology, and even mind-bending microbial ecology. In other words, this paper has something to interest everyone. So how should we begin? Let's start by asking what we'd like to learn. Well, here, the authors offered the following in the opening line of their abstract that well sets the stage. Transient or labile molecules in the GI tract, such as reactive oxygen species, often abbreviated ROS, and nitric oxide, which they abbreviate RNS, and hydrogen sulfide are key signaling molecules and mediators that suggest inflammation is taking place in our gut. And we know inflammation is bad. It often leads to disease processes. And this is one of these great papers that has a lot of things in their extended data section. And figure 1 of their extended data, if you're not familiar with what these reactive oxygen species do in a disease process like inflammatory bowel disease, they offer a quick review of the working hypothesis behind inflammatory bowel disease and how its severity is actually mediated by these labile molecules that are not all that easily detected in real time. And principally what happens in IBD is you get an inflammatory insult. You then get a disproportionate immune response via all the cytokine signaling that we try to forget. And then you get the release of these redox active molecules, such as the ROS or nitric oxide. And then that resulting oxidative stress inhibits microbial growth in the gut lumen. However, chronic intestinal inflammation then damages the epithelial layer that can lead to local invasion that then destroys the epithelial barrier, allowing intestinal microbes not only to invade the mucosa, but they can go systemic. Now, they also, on past TWiMs, we have discussed the role of thiosulfate and tetrathionate and hydrogen sulfide, all in this process. And we've also discussed enough about hemorrhagic diarrhea and ulceration of the colon. And all of these things are because of this reactive oxygen species. But these mediators of disease are labile. They're transient. They can go poof. And they cannot be measured very easily in real time with existing technology. As you might imagine, absent some sort of detection method, and owing to their highly reactive nature and extremely short half-lives in the human digestive tract, such molecules are difficult to detect until the pathology is evident. And the way we typically detect that pathology is through an invasive procedure like a colonoscopy or endoscopy, where they stick a tube either up your butt or down your throat. So today, we're going to explore how the authors developed a miniaturized device that integrates genetically engineered probiotic sensors in concert with a custom designed photodetector and communication system all tucked in together to track these molecules in our gastrointestinal tract. And you're probably saying, did he say photodetector? Yep, you betcha I did. We have previously introduced folks on TWiM to the notion of bioluminescent reporters, that when the genetic circuitry encounters a particular stimulus or an effector molecule of the circuit, the equivalent of these genetic transistors flip and emit light. And as a result of the reporter molecule being manufactured by the microbe in response to either the effector molecule or the stimulus detected by the bacterium, that was in turn activated by the genetic element which was responding to the perturbant. But the problem is, as you know, it's dark in our guts. So what they did is they leveraged the molecular specificity of these living sensors, or should I say now transistors, because we're really delving into electronics now. And they are able to encounter the labile molecule that they were attempting to measure. So, for instance, the signaling molecules like these ROSs or RNSs suggest inflammation. So what they simply do is they emit light. So how do you visualize light in the closed system of our digestive tract in real time? Well, here they took a low-powered electronic circuit that has a detector that will actually communicate that it's seeing light. And then they encapsulate this engineered microbe into this electronic device that then emits a wireless signal. So any of you wearing a continuous glucose monitor like the Libra 2 or the Libra 3 or one of the Dexicom products like the one Nick Jonas demonstrated during the Super Bowl commercial of 2023, where the sensor communicates directly with your smartphone, enabling you to measure in real time your circulating glucose concentration without the need for a needle stick. But from the perspective of today, wonder what such technology could afford a patient, or in this case, a scientist, trying to understand and then develop mitigative steps before the substantial pathology ensues. Now, all of this sounds great if true. So the data they're going to offer us shows us that not only does this technology work in monitoring the gastrointestinal tract in both small and animal models, but it's likely going to work in people. What's more is that they integrated these components into a device where it's encapsulated as they've taken bioengineered bacteria, they've taken an electronics, albeit a microelectronics, that they designed for appropriate swallowing. So this is a really small pill. It's 8.5 millimeters by 14.25 millimeters, and it has a total volume of about 1.4 cubic centimeters. It's a pill. So we're talking about something that you could easily swallow, that has been digested, that can actually pass through our gut without clogging the plumbing. And it's designed to answer the question many New Yorkers or many New Jerseyans seemingly are always asking of each other, how you doing? And I'm sure Vincent's heard that phrase much on the train. How you doing?
Oh, yeah.
And many microbiome related conditions, notably this inflammatory bowel disease, are, of course, associated with chronic intestinal inflammation. So it would be great if you could monitor whether or not you're having a flare, and then you would know to take your medication to settle the flare down by taking some of these drugs that they have developed for IBD. But oftentimes by the time you take them, the flare has flared, the immunology has taken place, and you're ending up with pathology. So as these molecules are labeled, you just can't mail in a stool sample. You can't run quick to your doctor to have it checked, because by the time you get there, it's going to be like taking your car into the car dealer where you say the car's not running. And of course, when you get it there, it's perfectly fine. So we need something in real time.
So it's the idea that you would take these when you feel like you are having a flare, because obviously they're only going to be transiently in you. So
right.
Okay. You can't monitor it that way unless you take them every day. But for instead of doing a colonoscopy or something, you could do it.
Right.
I just want to make sure.
Or they could have a retention time, how long it takes to transit through the gut system. Yeah. And I'm sure they're beginning to work on all of that. But here, this paper is principally proof of principle that they're trying to understand what's going on. And let's face it, today, understanding what's going on in the gut. And this is where the microbial ecology comes in. And this paper is going to get the microbial ecologists really excited, because we can make reporters to almost anything that's going to respond to a particular stimuli. And here they're just looking at something really straightforward, like reactive oxygen species, hydrogen sulfide, tetrathionate, things along those lines. They justify constructing the readout sensors, offering that these engineered bacteria could react to the local environment or stimuli. It's not only possible, but it's relatively straightforward to biologically engineer. And we really don't have all that great of a handle on what can trigger an IBD episode. We don't know what actually does it. Could it be stress? Could it be something in your diet? Could it be a virus? I mean, we just don't know the precise mechanism that can send you down this path where the IBD sufferers are going to indeed have a flare. So in their first experiment, they leveraged the promise of transient biomarker panels by combining these natural protein-based sensing elements for nitric oxide, hydrogen peroxide, tetrathionate, and thiosulfate. And we've covered this on a past TWiM where these genetically encoded elements actually can serve as memory circuits. And they did their first proof of principle that they could do this in the probiotic bacterium E. coli strain Nissl. And they validated its function in vivo in both the small animal model of the mouse and in the pig model, where they actually are using a model of inflammation. So in step one, they validated the circuitry/genetic element, where they're effectively testing whether or not the transistor will actually react to the physiological concentrations of their model system. So for the mouse model, they use the dextran sulfate to introduce this inflammation, and for the pig, they use an ischemia reperfusion model to induce inflammation. So they first optimized the nitric oxide sensor by combining the bacterial nitric oxide biosensor, which is NOR-R, with a DNA recombinase core circuit to create memory circuits that could record, but they still had to recover feces and then do a GFP fluorescence assay on it. It was a really cool experiment, and that's in figure 2A, because as the bug is transiting the gut, it's recording the information in the DNA that can be passed from generation of bacterium to generation. And we did that on a previous TWiM, but I couldn't remember which TWiM it was. So I'm going to have to do a search and find it for Ray to put into to the show notes. And then using the flow cytometry, the percentage of cells that were GFP on was calculated at each concentration of diethylamine, triamine, nitric oxide addict, which is the data no. And after multiple rounds of optimization to improve the signal to noise ratio of the genetic circuits and characterization, their recombinance-based system was able to respond to a concentration threshold of approximately 30 micromolar nitric oxide with high specificity and was not influenced by other reactive nitrogen oxide species present in the gut environment. This nitric oxide sensor was then tested under anaerobic conditions, and they built their memory recombinase circuits to detect hydrogen peroxide, thiosulfate, tetrathionate. And if that were not enough, they also created a disease stage detector based on the concentration of nitric oxide detected that will report whether inflammation in the gut was mild, moderate, or severe.
That's amazing. Amazing.
And this is all in their extended data. This paper has a lot of stuff in the extended data section. So take a look at it, because if you're curious about anything about the biology, it's buried in the extended data. They were enamored with the electronics, which we're going to get to here in a second. But it's so cool. So some of you might recall that we talked about building memory circuits. So now we have the ability to detect. We know the detector works. The circuit diagram is beautifully depicted in the insert of their figure 2. So you can see how the circuit will work, how the nitric oxide does its thing. So you get the idea of how this sucker is going to work. They validated it also in pigs using living pigs, and they're doing surgery on the pig, and they're implanting these devices in, and these data are shown in figure 2C and D. And here in the living pigs, using this reperfusion injury model of intestinal inflammation, they inject the bacterial biosensors directly into the lumen of the sedated pig in either the inflamed segments or the healthy segments, with or without the added biomarkers. And the nitric oxide sensing bacteria detected different concentrations of the nitric oxide in the control group, where they're physically adding the nitric oxides, and emitted a positive signal in some of the animals in the disease group, where they're mimicking natural inflammation by the ischemia reperfusion. So they're really actually testing if this will work in a disease state. And pigs are similar to humans in their digestive process. And so consequently, they're if you will, rapid prototyping from a small animal model to hopefully someday humans. Now, although these previous direct nitric oxide measurements in the inflamed intestines were limited and uncertain based on their lowest detectable signal from their nitric oxide biosensor, which was around 8 micromolar, here their results agreed with the estimated range of the expected nitric oxide concentrations of between 1 and 10 micromolar in humans, and suggests that such biosensors can be functional in the complex dynamic environment of an inflamed human intestine. But we still are unable to answer the question of how are you doing in real time. So this takes us to their third figure. And as you might imagine, the third figure describes how they created the biosensor that could leave the lab and leave the need for this invasiveness and to enter the real world of the clinic. Here they specifically designed a system that had the size and form factor that you could swallow it easily and won't get stuck in our plumbing. It's a non-deformable dosage, and they developed an optimized luminescent readout via this custom microelectronic bioluminescence detector, and they have sandwiched on top of this bacterial chambers that can actually hold these individual engineered bacteria that are going to react, flare some light, transmit the light to the detector that then can talk to something like, here they didn't use an iPhone like Nick Jonas does. They used a recording device in the lab. But you sort of get the idea of how these newfangled sensors make it out of the lab and into something like your arm, or in this case, we're going to swallow a pill. So basically, the bacteria are precisely aligned in the tablet in this 2x2 array with the microelectronic photodiodes, and the hermetic seal was maintained around the electronic components. So this enabled a sensing form so they can use their engineered microbes that have this reporter construct, and they then integrated this. And so the membrane has to be porous enough to allow these small molecules to come across. So the membrane is actually 0.4 micrometers in diameter. So this will allow these small molecules to freely diffuse back and forth. And the membrane didn't interfere with the detection of the target molecules when the chambers were placed in feces. And that's in their extended data in figure 6A. Additionally, to protect the onboard bacteria during ingestion and transit through the stomach, they also developed freestanding enteric polymers akin to many of the pills that you and I take so they don't dissolve in our stomach by the acid. So they have these enteric coatings on them. And they then generated versions of the bacterial sensors that can optimally couple their photosensitive electronics that replicated the GFP readout in this self-contained bioluminescent reader. So now you're going to say, does their gizmo
I have
work?
to say, this is like more of an engineering problem than a microbial problem, because engineering the bacteria is fairly straightforward. But this figure, I think it's is it figure 2 or figure 3?
It's figure 2 shows you how to engineer the bacterium. And any first year graduate students, that's one of their early projects. Make me a reporter
Exactly.
cassette.
But figure 3, where they show you the device, this pill.
Oh, yes.
It's nuts.
It's wild. It's nuts. It's truly nuts. And what they're measuring is current, and it's in the femtoamp range. So those of you who don't know what a femtoamp is, it's 10 to the minus 15 amps. You could never power your cell phone on that little juice. I mean, they have really sophisticated electronics. So they take us to the clinical region. So the bacteria are aligned in this device, and now we're getting to is, does the gizmo work? And that's in figure 4. So we're back into pigs, and we're again using the ischemia reperfusion model to induce inflammation. They did the surgery on the pig because they didn't trust the pig not to chew the pill to bits. And their electronics, they actually show in figure C, they show a little iPhone to detect the signal. And their design enabled the remote monitoring of these biomolecules in the gut for up to 4 hours with a 70 femtoamp relative photocurrent in the induced compartment and a negative 50 femtoamp in the control or the uninduced control compartment. There was also a lag of tetrathionate sensing, which is a real good indication of intestinal inflammation. And if you recall from some of our type 3 secretion papers, when we were talking about tetrathionate can actually feed salmonella, and that's what causes the salmonella bloom. So tetrathionate is a good marker to indicate local invasion and inflammation. So what these authors have done in their data is they showed that there is both specificity and sensitivity in their sensor. And you don't have to know much about the electronics. The one that's in the inflamed environment, it's going up, and the one in the control environment is just, sitting there. And so it's really quite remarkable that what the authors have done is they've built a microbial biosensor that is compatible with ingestion and can sense an array of biomarkers as they are being produced. Now, the authors didn't go into this in the discussion, but I immediately went to all of those papers we talk about with the microbiome in understanding whether probiotic organisms are doing what they're supposed to do. And the thing that we all need the bacteria in our gut to do is make butyric acid. Except butyric acid is consumed by everyone. And so if you're taking a probiotic, you're going to want to know if that creature is making butyric acid for you, even if everybody else is eating them. So if you put one of these sensors in, you can actually demonstrate. And this is where the microbial physiology comes in. You can engineer these things based on what we're learning about the microbiome. So this is a real way of getting these molecules into small animals and larger animals and even people to begin to decipher what's going on in the microbiome. Now, the authors, this is me purely speculating of where this will go next, but this is a really fantastic paper. They integrated these bacterial sensors with these custom designed integrated photodiode chips. And the question that, I ended my thought on this was, are these chips going to make irrelevant the endoscopists of the world?
I don't think they're going to do that, only because what they can biopsy in an endoscopy and they can see precancerous lesions. Yes. And these can only sense. This is like taking the blood test you would take to detect like prostate cancer or something. It can't actually confirm it like a biopsy could. The question is, would they, if this works really well, then they might be able to get rid of some of the sort of preventative endoscopy procedures.
Yes.
But in the end, if there's any signal, you're going to have to do an actual endoscopy. Is my guess.
But the thing is, you could do this more frequently.
Yes. Right? It doesn't does it require prep? It's all about the prep, right?
Yeah, it's all about, it's got to be clean in there. Otherwise, you get obscured. Yeah. Because then the prep is horrible. So,
well, this is something that I thought our listeners would enjoy looking at. It
is. It's very cool.
And it's all based on those simple reporter cassettes that any undergraduate class is using ubiquitously.
Yep.
I mean, Mark Martin loves his bacteria that make light.
He does. He talks about them all the time. He sends me stuff. Well, they're not bacteria, but they're luminous.
They're luminescent, yes.
All right, Michael, thank you very much. That is TWiM 297. You can find the show notes at microbe.tv/TWiM. You can send questions or comments to twim@microbe.tv. And if you enjoy our work, we would love to have your financial support to continue this. You can go to microbe.tv/contribute. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levin is at Washington University in St. Louis. Thank you, Petra.
Thank you, everybody.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.